After studying this module, you shall be able to know about DNA extraction procedure, learn the purification process of DNA and how DNA is fragmented. Introduction. Physical evidence cannot be wrong, it cannot fabricate itself, it cannot be wholly absent. Only human failure to find, study and understand it can diminish its value, as quoted by Paul Kirk, Crime Investigation, 1953. Prior to a DNA test can be performed on a sample, it must be gathered, separated and kept in the best possible condition for further interpretation. This part covers the critical points of DNA extraction, purification and fractionation. Technique for segregating and making copies of sequences of DNA is essentially utilized as a part of DNA fingerprinting or DNA typing. The technique was created in 1984 by the British geneticist Alec Jeffreys after he saw the presence of specific groupings of DNA called mini satellites that does not contribute towards the gene function, but gets repeated in the gene and also in other genes of the sample of DNA. The procedure for making a DNA fingerprint comprises of numerous steps, that is, first requirement is the sample of cells containing DNA, for example from skin, blood or hair, then separating the DNA and at the end purifying it. DNA is subjected to restriction enzymes called as endonucleases. The restriction enzymes cut the DNA into pieces. The restriction enzymes are of different types, for example, EcoR1, PST1, HinF1, etc. When DNA is subjected to the enzymes, the enzyme recognizes a particular sequence and cut these sequences in one or two or more fragments. These restriction fragments are then separated by gel electrophoresis. The gel electrophoresis separate the pieces of DNA based on their size. These fragments migrate towards the positive electrode, and in this the smaller fragments move faster than larger fragments, thus separating the DNA samples into bands. These double stranded fragments are then denatured by high alkali treatment into single strands. These single stranded separated fragments are then transferred or blotted to nitrocellulose filter or nylon membrane. Next step is to make these single stranded fragments into double stranded fragments. This process of making single stranded into double stranded is known as hybridization. This is done by addition of probe to the nylon membrane or nitrocellulose filter. A probe is single stranded DNA fragment or synthetic DNA and is stacked with radioactive marker such as P32. When the probe DNA mixes with single stranded fragments on nylon membrane, the probe will attach to particular segments, that is complementary sequence. Thus, the attached fragment will be radioactive. The nylon membrane is then washed with 0.05% STS, and in this process the loose probe is removed. Then the nylon membrane is put in contact with X-ray film, which is then exposed and developed. The X-ray films show grey to black bands. These bands represent hybridized radioactive probe with complementary sequences. DNA extraction. The biological material encountered most often at scenes of crime is blood. This is mainly because of the violent nature of many crimes and also because it is easier to visualize than other biological fluids such as saliva. Other frequently encountered samples include seminal fluid, which is of prime importance in sexual assault cases. Saliva that may be found on items held in the mouth, for example, vessels used for drinking and cigarette butts or on the bite marks, and epithelial cells deposited, for instance, as dandruff and in feces. With the increase in the sensitivity of DNA profiling, the recovery of DNA from epithelial cells shed on touching has also become possible. Hairs are naturally shed and can also be pulled out through physical contact and can be recovered from crime scenes. Naturally shed hairs tend to have very little follicle attached and are not a good source of DNA, whereas plucked hairs or hairs removed due to a physical action often have the roots attached, which is a rich source of cellular material. The three stages of DNA extraction can be classified as disruption of the cellular membranes resulting in cell lysis, protein denaturation, and finally isolation of DNA from the denatured protein and other cellular components. Some of the extraction methods usually applied in forensic laboratories are described. Chilex 100 resin. Chilex 100 is a resin that is made up of styrene divinyl benzene copolymers comprising combined immunodiacetate ions. The resin has a great attraction for polyvalent metal ions like magnesium. It chelates the polyvalent metal ions and efficiently eliminates them from solution. The drawing out process is very easy. The Chilex R100 resin, which is delivered as beads, is made into a 5% suspension using distilled water. The cell matter is incubated with the Chilex 100 suspension. Proteinase K, which digests most cellular protein, is often added at this point. This incubation is monitored by 8 to 10 minutes at 100 degrees Celsius to confirm that all the cells have ruptured and that the protein is denatured. The tube is then simply centrifuged to pellet the Chilex 100 resin and the denatured protein at the bottom of the tube, leaving the aqueous solution containing the DNA to be used in PCR. The pH of the Chilex suspension is between 9 to 11 and hence it is alkaline in nature, and because of the single stranded DNA is obtained by this procedure. Silica based DNA extraction. Usually the salting out process has been widely employed in the field of molecular biology. The very first step in the extraction process comprises of incubating the cellular material in a lysis buffer, which includes a detergent and proteinase K. The commonly used detergent are sodium dodecyl sulfate, tween 20, triton X100 and nonidate P40. The cellular structure breaks down because of the destabilization of the cellular membranes by the lysis buffer. The protein structure is disrupted by interference with van der Waals interactions, hydrogen bonding and the hydrophobic interactions because of the addition of a cheotropic salt such as 6-M guanidine thiocyanate or 6-M sodium chloride during or after cell lysis. The reduced solubility of the cellular protein is caused by the excess of ions in the high concentration of salt competing with the proteins for the aqueous solvent, effectively dehydrating the protein. DNA will bind to silica or glass particles with a high affinity in the presence of a cheotropic salt. Release of DNA from silica or glass particles is carried out by suspending them in water after the removal of other cellular components. Without the cheotropic salt, the DNA no longer binds to the silica or glass and is released into solution. Phenol chloroform based DNA extraction. Earlier, the phenol chloroform extraction technique has been widely employed in the field of molecular biology, but its use has been slowly phased out in the mid of 1990s, majorly owing to the toxic nature of phenol. Cell lysis is performed as in the previous method. Phenol chloroform is added to the cell lysate and mixed, and the phenol denatures the protein. Centrifugation of the extract is carried out and the protein precipitates results in the formation of a pellicle at the junction of the aqueous phase layer and that of organic phenol chloroform phase. The process is repeated 2 or 3 times or until there is no visible pellicle. The DNA is filtered out of the aqueous phase by ethanol precipitation or filter centrifugation. FTA paper. FTA paper was applied for sample gathering and storing mainly from buccal swabs and fresh blood samples. Once a sample is applied to the FTA paper, it is stable at room temperature for several years. Cellular materials lyses on contact with the FTA paper and the DNA becomes bound to the paper. To analyze the DNA sample, the first step is to take a small region of the card, normally a 2 millimeter diameter circle, place it into a 1.5 ml tube and the non-DNA components are simply washed off, leaving only DNA on the card. The small circle of FTA paper is then added directly to a PCR. The FTA paper extraction are very simple to perform and do not require multiple tube changes, thus reducing the possibility of sample mixing. Purification of DNA from cell extract. In addition to DNA, the cell concentrate will also contain protein and RNA. The purification methods can be utilized to remove these contaminants, leaving the DNA in a pure form. To remove contaminants from the nucleic acids, the frequently used method is solvent extraction. For example, a combination of phenol and chloroform is frequently used to remove proteins. For concentrating the nucleic acids, precipitation is usually carried out with ethanol or isopropanol. To increase the precipitation efficiency of a product mixture, inert carrier, for example of glycogen, can be added. In case when the amount of target acid nuclei is low, various other precipitation techniques for nucleic acids comprises of precipitation of proteins involving changes in pH and selective precipitation using high concentration of salt. This is salting out. Techniques of chromatography may utilize various separation techniques such as selective adsorption, ion exchange, gel filtration or affinity binding. Gel filtration exploits the molecular sieving properties of porous gel particles. A matrix with defined pore size allows smaller molecules to enter the pores by diffusion, whereas bigger molecules are excluded from the pores and eluted at the volume. Thus, molecules are eluted in order of decreasing molecular size. Another technique which utilizes electrostatic interaction between a target molecule and functional group on the column matrix is ion exchange chromatography. Nucleic acids characterized by high negative charge and a linear polyatomic structure with the help of simple salt buffers can also be eluted from ion exchange columns. In all adsorption chromatography, nucleic acids adsorb selectively into silica or glass in the presence of certain salts. For example, cheotropic salts. while other biological molecules do not. A low salt buffer or water can then elute the nucleic acids, producing a sample that may be used directly in downstream applications. Selective centrifugation is a powerful purification method. For instance, a plasmid purification, ultracentrifugation in self-forming CSCl gradient have been employed at high G forces. Frequently, centrifugation is combined with another method. An example of this is spin column chromatography that combines gel filtration and centrifugation to purify DNA or RNA from smaller contaminants, salts, nucleotides, etc., for buffer exchange or for size selection. Some procedures combine selective adsorption on a chromatographic matrix with centrifugal elution to selectively purify one type of nucleic acid. Fragmentation of DNA. DNA fragmentation plays an important part in forensics. Restriction fragment length polymorphism or RFLP is a technique of breaking the variable lengths of DNA pieces that results from processing a DNA sample with a restriction endonuclease. When DNA is subjected to enzyme, the enzyme recognizes a particular sequence and cut these sequences in one or two or more fragments. These restriction fragments are then separated by gel electrophoresis. These single stranded separated fragments are then transferred to a nitrocellulose filter or nylon membrane. This transfer technique is called as Southern blot after E.M. Southern, who first described the technique. Next step is hybridization, that is, making single stranded fragments into double stranded fragments. This is done by addition of probe to the nylon membrane or nitrocellulose filter. It can differentiate two samples originating from different sources using fewer loci than other system. Determine more readily whether single sample contains DNA from more than one person. Power of discrimination is more due to hypervariability at each locus and ability to look at many loci. In polymerase chain reaction analysis, the DNA is extracted from samples and the extracted DNA is mixed with short fragments of known DNA called primers. It is three-step procedure. In first step, DNA is extracted and denatured to form single stranded DNA. In second step, this single strand DNA is mixed with single strand primer DNA. In the last step, that is third step, DNA is synthesized by primer extension from 3' end to a 5'. At the end of third cycle, identical double stranded DNA appear. It requires only trace amount of DNA and procedure requires less time. It is highly sensitive method. Now, let us summarize what we have learnt in this module. The procedure for making a DNA fingerprint comprises of numerous steps, that is, we need sample of cells containing DNA, then separating the DNA and at the end purifying it. With the increase in the sensitivity of DNA profiling, the recovery of DNA from epithelial cells shed on touching has also become possible. The three stages of DNA extraction can be classified as disruption of the cellular membrane resulting in cell lysis, protein denaturation and isolation of DNA from the denatured protein and other cellular components. FDA paper was applied for sample gathering and storing mainly from buccal swabs and fresh blood samples. Once a sample is applied to FDA paper, stable at room temperature for several years. cellular material lyses on contact with the FTA paper and the DNA becomes bound to the paper. Restriction fragment length polymorphism is a technique of breaking the variable length of DNA pieces that results from processing a DNA sample with a restriction endonuclease. In polymerase chain reaction analysis, DNA is extracted from sample and the extracted DNA is mixed with short fragments of known DNA called primers. It requires only trace amount of DNA and procedure requires less time.